Human reproduction relies on sexual dimorphism, requiring the precise coordination of two entirely separate anatomies to produce a single viable offspring. This process functions as a continuous, failure-intolerant biological pipeline, running from the initial cellular division through to postnatal nourishment. This pipeline consists of 7 discrete stages: gametogenesis, insemination, fertilization, implantation, gestation, parturition, and finally, lactation. The pathway crosses multiple orders of magnitude, scaling from the microscopic division of haploid gametes to the macroscopic physiological changes required to deliver a fully formed human. Advancing from one step to the next requires a series of biological switches, consisting of highly synchronized neuroendocrine signals that trigger each phase at a precise physiological moment. The male primary sex organs, the testes, operate under a strict thermal constraint. To produce viable sperm, they must maintain a temperature roughly 2 to 2.5 degrees Celsius below core body heat. Internally, the testes are divided into approximately 250 compartments called testicular lobules. Each lobule contains 1 to 3 seminiferous tubules, which serve as the actual manufacturing sites for sperm cells. This is the hypothalamic pituitary gonadal axis, or HPG axis, the system that initiates production. The hypothalamus releases gonadotropin-releasing hormone, or GnRH, which signals the anterior pituitary gland to release 2 distinct gonadotropins. These signals bifurcate into luteinizing hormone, or LH, and follicle-stimulating hormone, or FSH. LH stimulates the Leydig cells to produce androgens, like testosterone, while FSH targets the Sertoli cells to manage gamete maturation and provide essential nutrients. Under this endocrine control, spermatogenesis begins. This is a sequence of rapid cellular division where a single parent cell splits to produce multiple reproductive units. The meiotic sequence begins with a diploid spermatogonium, a cell containing the standard 46 chromosomes, undergoing mitosis to become a primary spermatocyte. This cell then executes 2 rounds of meiotic division, reducing its genetic payload to form 4 haploid spermatids, each carrying 23 chromosomes. These spermatids then undergo spermiogenesis. They physically transform, developing a head to house the nucleus, a midpiece packed with energy-generating mitochondria, and a flagellar tail for propulsion, resulting in motile spermatozoa. Male gametogenesis operates as a continuous manufacturing loop, strictly dependent on thermal regulation and neuroendocrine feedback. The female reproductive tract links the primary sex organs, the ovaries, to the uterus via the fallopian tubes. This lateral cross-section shows the pathway the gamete must navigate. The uterine wall consists of specific functional layers: the myometrium, a thick layer of smooth muscle, and the endometrium, a highly vascularized lining that thickens and sheds in preparation for gestation. Within the ovaries, the tissue is divided into an inner medulla containing blood vessels and nerves, and an outer cortex where gamete maturation occurs. Follicular development occurs in the cortex. This cyclical timeline shows an immature primary follicle layering on new cells and expanding an internal fluid cavity to become a mature Graafian follicle. At its peak, the Graafian follicle ruptures, ejecting a single haploid secondary oocyte. The remaining tissue collapses and transforms into the corpus luteum, a temporary endocrine structure. The released oocyte is swept into the infundibulum and rests in the ampulla, the widest part of the fallopian tube. This system manages 2 simultaneous mandates: the maturation of a haploid gamete and the construction of a nutrient-dense uterine environment to sustain potential gestation. To exit the male body, spermatozoa travel from the rete testis through the epididymis and up the muscular vas deferens. 3 accessory glands, the seminal vesicles, the prostate, and the bulbourethral gland, inject fructose for energy, calcium, and lubricating mucus into the transport line. This combined seminal plasma, mixed with sperm, forms semen. Following insemination, the spermatozoa must navigate the female tract to reach the secondary oocyte, specifically within the ampulla of the fallopian tube. At the precise point of contact, a single spermatozoon impacts the thick outer protective membrane of the oocyte. This initiates the acrosomal reaction. The spermatozoon's head releases specialized hydrolytic enzymes that dissolve the oocyte's barriers, allowing the two cells to fuse their membranes. Fertilization constitutes a specific enzymatic sequence, requiring precise spatial and temporal alignment to successfully merge two sets of genetic data. After fusion, the single-cell zygote begins cleavage, executing rapid mitotic divisions as it travels down the isthmus toward the uterus. These divisions result in a hollow, multicellular structure called a blastocyst. This microscopic cross-section shows implantation, where the blastocyst physically invades and attaches to the vascularized uterine endometrium. During the 9-month gestation period, this embedded connection facilitates the massive transfer of nutrients and oxygen required for fetal development. During parturition, the smooth muscles of the myometrium transition into a phase of rhythmic, high-force contractions to expel the fully developed fetus from the uterus. The uterus shifts its mechanical role from a stable, passive incubator to a high-force engine of expulsion. This clinical schematic reveals the internal architecture of the mammary gland. Milk is produced in clusters of cells called mammary alveoli before moving through the ductal pathways to the nipple. The hormone prolactin drives the production of milk by acting on the alveolar epithelium. While prolactin drives production, the separate hormone oxytocin triggers the myoepithelial cells to physically contract, squeezing the alveoli and actively ejecting the milk through the duct system. The milk produced in the first few days, called colostrum, contains a high concentration of IgA antibodies. IgA, or immunoglobulin A, is an antibody that provides the infant with essential, immediate immune defense. Biological reproduction continues beyond birth, utilizing distinct neuroendocrine loops to secure the offspring's early survival. To propagate the species, biological matter traverses a sequence of spatial checkpoints: the testes, the ampulla, the uterus, and finally the mammary glands. Each phase depends on the mathematical accuracy of meiotic reduction followed by flawless neuroendocrine signaling. Small failures at the microscopic level prevent macroscopic outcomes. The survival of the organism is built on the precision of these hidden cellular mechanics. Human reproduction is a strictly engineered biological chain linking a single cellular division to the propagation of the entire species.